Okay, so I again did it. Thank God this is the last chapter. And I kind of cut myself off there, and I realized I wasn't watching the time. I'm sorry. But anyway, picking up there, our photoreceptors are called rods and cones. And cones, color, see for color, right? Rods are more sensitive, but it won't send an actual potential that decodes in color. Not it's not sensitive to individual wavelengths of light, because that's what color is. Okay. Well, whether it's a rod or a cone, they're going to synapse with these bipolar cells, because these are specialized receptors. And so here's our bipolar neurons again, like in the olfactory, and these will connect to ganglion cells, which then again, cell bodies, collection of cell bodies outside the central nervous system, are going to then transmit the action potentials through the optic nerve. It'll leave as the optic nerve. And there's a couple other cell types in here that can help modify this, but eventually all these axons collect together at the optic disc. And so at the optic disc there are no rods or cones, and we also call this the blind spot, because there's no way, if light hits the optic disc, there are no receptors to provide vision. And this is, of course, where it leaves as the optic, collectively as the optic nerve. All right. So other features, again, we've already mentioned the optic disc, but I'll reinforce it here, right? Blind spot, no photoreceptors, only axons. So all of these ganglion cells are sending their axons, and they all collect or converge right here. Photoreceptors are actually behind axons. Let me see, back here, right? So these retinal ganglion cells, right, axons that will collect at the optic disc. So here, as we look at this, what you see is light is actually transmitted in, excites these guys, and then they synapse through these layers of cells into these ganglion cells. So here I'm saying that, right, photoreceptors are behind the axons, right, the retinal ganglion cells, bipolar cells, and blood vessels, which absorb some of the incoming light. Okay, at the exact center of the retina, so where if light was coming in from a distance, right, and it would come in parallel waves, we would find this region, which is called the fovea centralis. And the overall region is actually called the macula lutea, so this is the spot. And in the middle, central, right, of that spot is this little depression called the fovea centralis. And we can actually see this on the real eyes, and we'll look at this on the slides. This area contains only photoreceptors, and so therefore it gives us the best or the sharpest vision. Okay, so everything, light is being optimized by all these other structures so that the majority of it will strike right here. Okay, and that's, again, remember, vision is our survival. All right, so these visual receptors, rods and cones, just to add a little bit more to this, right? So light falling on the retina changes the pigment molecules, which then cause a change in the activity of those retinal ganglion cells. All right, so two segments, there's an inner, right, so here we're really looking up close at a rod cell, two segments. Inner has a nucleus and organelles, outer has a photoreception due to these pigments. All right, so these are specialized proteins. All right, so here's just looking again at what a rod cell would look like, and here's the other drawing of this. So the pigments contained by the rods is called rhodopsin, and cones have these three different opsins. And those cones then those different opsins are sensitive to specific wavelengths of light, which is why they help us see in color. Right? So light-sensitive pigments, again, rhodopsin or opsins, that then decompose when exposed to light. So here again, a change has to occur. So rhodopsin in the rods, which decomposes to opsins and retinol, right? And so this is why we need vitamin A. It helps us construct this pigment known as retinol. In the darkness, and we need vitamin A for other things too, but this is one of the reasons we need it. In darkness, it turns out sodium channels are open. And so in light, the rhodopsin, right, this pigment, will absorb light, and this changes the shape. So this light energy changes the shape, and they degrade, they fall apart, and this causes sodium channels to close, which results in a hyperpolarization. Okay? And the more intense the light, the more they hyperpolarize. And so in light, it turns out rods don't work, right? When it's dark, if it's completely dark, nobody's working because there is no stimuli, there's no light, right? But in these times when there's less light, which is what we're talking about, sodium channels are open and these rods are functioning. when it's very bright out, rods stop functioning, okay? And we're going to illustrate this in a second here, but if you're like confused by this. So why do you see when it's light? Well, because your cones continue to work. So the rods are actually hyperpolarized, and we call this a bleaching effect, right? And so I'm sure you can relate. In bright light, most of the rhodopsin is decomposed, so you have reduced sensitivity or bleaching of the rods. But the cones keep functioning, and we see everything in color, right? In dim light, rhodopsin can be regenerated from the opsin and retinol, and this does require energy to put this back together. The cones, however, are unstimulated, right? They're not getting enough energy, and so things appear not in color, right? In shades of gray. So how do where's the in-between? Well, this occurs when we walk into a dark building from the light. If you go immediately from a very bright, I talk like a movie theater or walk into a closet, right? Well, it turns out when you walk into the dark, of course, the cones stop working. And your rods have not been working, so they need some time to generate the rhodopsin. So in that a few minutes as we begin to adjust, right, where first you walk in and it's like you can see nothing, and then suddenly there's shadows, and then you can make out some. Again, as long as there is some light when we talk about dark, not cave dark, right? Now, as I mentioned before, if you're completely deficient in vitamin A, you have night blindness, so you can't regenerate the rhodopsin because you can't make the retinol. So that's clearly, but most of us do not have night blindness and have never experienced this, so we basically just need a little time, and we'll be able to adjust to the less light. If your retina is detached, of course, you also have blindness, but that's not just night blindness, that's also in the daylight as well. All right, the cones, they have pigments called idoopsins, and this is retinol and some protein, different proteins. You don't have to know their names, but there is, of course, some sensitive to red light, green light, and blue light. And color is basically the result of, getting combinations of these cones, different cones, excited, or starting to generate action potentials. And the combined action potentials of these different cones give us the range of color. And color, of course, is our perception in the primary visual cortex in the occipital lobe, right? So pathways for the impulses, again, here we have light coming in, passes through these nerve fibers from the ganglion cells, and strikes the rods and cones, all right, and gets them excited if it can, all right? Whatever doesn't hit them is absorbed. All right, these guys start an action potential, which is then transmitted through synapses to these bipolar neurons and horizontal cells. And there's these, American cells too that also help to modify. All right, so I say here rods and cones synapse with bipolar, and together with horizontal cells and amacrine cells, we modify, right? So we determine brightness and contrast with these cells. The bipolar and horizontal cells synapse with ganglion cells, and these axons converge in the optic nerve, cranial nerve 2. All right. So altogether we see because, and again, you want to be able to trace the pathway of light. Iris here, which is made up of radial and longitudinal muscles, so circular and longitudinal. Iris controls light entering. All right, so it's colored and muscle itself. The cornea lens, so cornea here and lens, and humors, right, will refract light, and so they bend it a little bit, so light coming in can come in as parallel waves, or it can be coming at divergent angles. So regardless, these structures help to refract light and try to direct it back here to the fovea. The retinal cells generate the action potentials. So we move in through the cornea, through the anterior chamber of the anterior cavity, and through the pupil, and hit there's a posterior chamber here, and then the lens, and then the posterior cavity with vitreous humor, and then the retina. All right. How is all this information delivered and processed? So the ascending pathways are tracks, and is dorsal, the column. The ascending pathway track, which is on the backside of your column, right? Your sensories are dorsal. And the spinothalamic tract both begin as neurons in the dorsal root ganglia, all right? So this is all, again, we're out of our special senses here, and we're thinking about how do we right, yet sensory. And then somatosensory, right, from the head and neck, use the trigeminal nerve. So again, we've got to bring all this sensory information up to the brain for us to have sensation, true sensation and perception. All right, so here's the dorsal column for touch and proprioception. All right, so here here's our spinal cord coming up through the brainstem, medulla, midbrain, and the cerebral cortex. All right, so there's this fasciculus cuneatus and the fasciculus gracilis, so upper and lower body, and I'm not going to ask you those names, all right? I'm not, okay? But this we want to. These are the actual pathways. So these are collections of these axons. They terminate in the medulla, where they synapse and are bundled together and come to the thalamus. So this is we see a lot of sensory comes up through the thalamus, with the exception of olfaction. Synapse is here and moves to a post-central gyrus, which again is called the somatosensory cortex. It's part of the parietal lobe. So they're just showing this kind of out of context here as we have a slice through here. They do refer to these as, the first order neurons, the second order, who it synapses to, and then the third order, but I'm not going to use those terms either. So it's just following the tracks. Okay? So again, fine touch, proprioception, pain and temperature come through these sensory, right? The spinothalamic tract, which is for pain and temperature, from the dorsal horn where the cell bodies, right, ascend to the thalamus, hence the name, right, from the spinal cord to the thalamus. From the thalamus, we will connect to the postcentral gyrus as well, right? So, again, same cartoon here, we're just focusing on this side. All right, so I will ask you, if I give you a name like spinothalamic tract, you should know that that is a sensory, and it's ascending, it's coming up. And that's because ascending tracts are generally named by where they start and where they end. And descending tracts are named the same way. So you should be able to discriminate from the name whether I'm talking about an ascending sensory or a descending motor response. All right, taste and hearing pathways. So gustation, your taste, and audition, your hearing, right? So taste is through the facial and glossopharyngeal, and they synapse, project to the thalamus again, right? And then to this gustatory cortex in the parietal lobe and part of the insula. Hearing comes in through the vestibular cochlear nerve, again through the medulla, combine to localize through there's part of the inferior colliculus, continues on to the inferior colliculus, which then projects to the thalamus again, and then sends it to the auditory cortex, which is in the temporal lobe. The superior colliculus receives information from visual and somatosensory and turns the head towards sound. So again, these are just, how do these function? We're just applying some of the things we learned from chapter 13 here in chapter 14 as we are trying to make sense of these senses or stimuli. All right? Balance is a vestibular system. So axons from the vestibular nerve, right, that were attached to the vestibule in the semicircular canals, bring information from these parts of the inner ear. They synapse with medulla and go directly to the cerebellum, some of them. Again, since it's balance, and balance is something that the cerebellum helps us figure out. There's a reticular formation, and there's influences on respiratory and cardiovascular in relation to body movement. So this is just going to tell you how complicated things can get. Or the spinal cord to initiate reflexes involved in posture and balance. So we don't even have to come up to the brain if we don't want to. They also project to the oculomotor, the trochlear and abducens, which compensates for head and body movements by stabilizing images on the retina, so that we, aren't left with a sense of vertigo and imbalance. So pretty complicated. Again, I'm not looking to drill you on these pathways. I just want to mention we should be aware of these and where we have to go in the brain in order to have this sensation and perception. All right, I'm going to stop before I run out of time here on this video. All right, and so we'll pick up on the last video and talk about our visual pathways.